In last class, we have studied biological oxidation. Biological oxidation, it is comprised of electron transport chain and mainly oxidative phosphorylation. So we have seen after digestion, the food macromolecules, they are transformed into simplest form, and mainly from the carbohydrate group, it is glucose. Main idea is to derive energy from the food particle. So we have seen after the digestion, the glucose is absorbed in the body, in the cell, then it is metabolized in different metabolic pathway, and thereby they produce reducing equivalents, especially NADH, and that FADH2. These reducing equivalents, they are oxidized in electron transport chain. In electron transport chain, during the time of oxidation, there is generation of proton gradient. So up to this much we have seen. Now the function of proton gradient is to produce ATP, and that is done by the complex 5 or ATP synthase. So we will see how it is done. Now, in last class, we have seen what is phosphorus. So, substrate level phosphorylation, where ATP is synthesized direct in the metabolic pathway from the high energy compound. From the high energy compound, the phosphate group is transferred to ADP and ATP is synthesized. And in case of oxidative phosphorylation, the reducing equivalent produces proton gradient, and the inorganic phosphate, using that proton gradient, whenever there is transfer of electron through the electron transport chain, there is release of energy. That energy is utilized for synthesis of ATP by ATP synthase. Here, inorganic phosphate is taking part in the formation of ATP. So, how it is done, we will see. Now, let's see the difference. This oxidative phosphorylation and your substrate level phosphorylation, these both are very important. It may be a short note. So, if there is short note in substrate level phosphorylation, you can write these points. In oxidative phosphorylation, you can write these points, and you can elaborate something from the next slides. So, phosphate group is transferred from substrate to the ADP. Whereas in case of oxidative phosphorylation, uses energy released from the electron transfer through ETC, electron transport chain, to generate ATP. Now, substrate level phosphorylation, it occurs in cytosol as well as mitochondrial matrix. Cytosol, for example, in glycolysis, Okay, 1,3-bisphosphoglycerate, when it is converted into 3-phosphoglycerate, the first phosphate group from the first carbon is used for synthesis of ATP. So, in case of glycolysis, it is in cytosol. In case of TCA cycle, tricarboxylic acid cycle or Krebs cycle, it is in the mitochondrial matrix. So, both in mitochondrial matrix as well as cytosol, this substrate level phosphorylation occurs. Synthesis occurs from directly from the metabolic pathway to the ATP. So, in case of substrate level phosphorylation, oxidative phosphorylation, it occurs in inner mitochondrial matrix. There are electron transport chain coupled with the ATP synthase, that is oxidative phosphorylation complex. Then it is a direct phosphorylation. This is indirect phosphorylation. Occurs in glycolysis and Krebs cycle and occurs in electron transport chain. So, oxidative phosphorylation has to occur in electron transport chain only. This is the glycolysis, pathway of glycolysis. Here, the 1,3-bisphosphoglycerate, when it is converted to 3-phosphoglycerate, ATP is produced. Here also phosphoenol pyruvate, when it is converted to pyruvate, ATP synthesized. Where see the amount of energy which is released during the hydrolysis of 1,3-bisphosphoglycerate and phosphoenol pyruvate. So, for synthesis of ATP, only minus 7.3 kilocalories is required, whereas so much calories released from the energy. So, this is a reaction suitable for synthesis of ATP. Here is the Krebs cycle, where GTP is produced, guanosine triphosphate. It is like a ATP only. From guanosine triphosphate, the ATP is synthesized. So, it is not, this ATP is not directly synthesized in this reaction, but ultimately it is the ATP which is generated from using this release of energy of this reaction. So, this is the world's smallest rotor stator molecular motor. This is the motor where the generation of power occurs. We know ATP is the powerhouse of mitochondria. Mitochondria is the powerhouse of cell, and in the mitochondria, power is generated in the form of ATP. And this is the motor which is being used in the generation of ATP. It is so small that you need to see this motor with the help of electron microscope. Okay. So, ATP is synthesized in complex number 5. Here are the electron transport chain, complex 1, complex 3, complex 4. These are used by the reducing equivalent NADH. FADH is not, I have not shown here. FADH is using only this complex number 3 and complex number 4. While electron is transferred through these complexes, the proton, in case of complex number 1, 4 protons are transferred. In complex 3, 4 protons are transferred. Again in complex 4, 2 protons are transferred. So, when electron from the NADH is transferred to the electron transport chain, it pumps through 10 numbers of protons in the intermembrane space. Okay. Now, if it is FAD, then it will not utilize complex 4, it will transfer the electron directly to coenzyme Q, then utilize these two only, complex 3 and complex 4. So, complex 3, it is capable of pumping 4 number of electron, 4 number of proton, and complex 4, it is capable of pumping 2 numbers of proton. So, in case of NADH, it is the 10 numbers of proton that is translocated in the intermembrane space, and in case of FADH2, it is the 6 numbers of proton which is translocated in the intermembrane space. Now, for synthesizing ATP, we need 4 numbers of proton. 4 numbers of proton. Now, to define oxidative phosphorylation, it is nothing but the transport of electron through electron transport chain when it is linked with release of free energy. Now, the process of synthesizing ATP from ADP and inorganic phosphate coupled with electron transport chain is known as oxidative phosphorylation. The site is inner mitochondrial membrane by complex 5. Complex 5 is not a component of electron transport chain. Okay? The component of electron transport chain are complex 1, complex 3, complex 4. Complex 1, complex 2, complex 3, complex 4. Complex 2 is used by FADH2. So this is the where it is synthesized from the inner mitochondrial membrane. The repeated folding is called cristae, and from there it is hanging inside the mitochondrial membrane as like of a lollipop. Okay. Now here it is synthesized. So in the mitochondrial matrix, ATP is synthesized and released, and from the mitochondrial membrane, ADP is taken up again for synthesis of ATP. So there should be adequate supply of ADP, and there should be this generated ATP should be removed from the mitochondrial membrane to the peripheral tissue for utilization. That is done by a protein called ATP-ADP translocase. This is an enzyme, the 7th group of enzymes, 7th group of enzyme translocase. No? So this is one of the example of class 7 of enzyme. We can classify into 7 group of enzymes, no? Oxidoreductase, then OTHL and T. So last group is 7th group is translocase. So this is the one example of translocase. So electron is transferred to FMN, then through iron sulfur complex to the coenzyme Q, and 4 numbers of ATP. 4 numbers of protons are pushed, pumped in intermembrane space. Then from coenzyme Q to cytochrome B, and through iron sulfur complex to cytochrome C1. During this oxidation process, again 4 numbers of protons are pumped in the intermembrane space. Likewise, in the complex 4 also, it is 2 numbers. So total 10 numbers of ATP in case of NAD and 6 numbers of proton in case of FADH2. And this proton gradient is used by the complex 4 to generate ATP, and for each ATP we need 4 protons to be passed through the complex number 5. Now we are using the inorganic phosphate. So one term is the PO ratio. PO ratio, it is a short note. It will come as short note. So you will write like that. How you will write? Give the definition first. This is number of inorganic phosphate molecules utilized for ATP production. Mention that using electron transport chain by oxidative phosphorylation. Okay. Number of organic phosphate molecules utilized for ATP production by oxidative phosphorylation for every atom of oxygen consumed. Okay. So we have seen NAD provides 10 numbers of proton, and each ATP to generate requires 4 numbers of proton. So with the 10 numbers of ATP, we can synthesize 2.5 number of ATP. So PO ratio of NAD is 2.5, and PO ratio for FADH2 is 1.5. So first you will give the definition, then explain how many ATPs are synthesized, how many protons are pumped out in the intermembrane space by NADH, and how many protons are pumped out in the intermembrane space by FADH2. Okay. Now for synthesizing ATP, how many protons are required, and so how many ATPs are generated by the NADH, and how many ATPs are generated by FADH2. Okay. So mention. So that is the number of ATP generated by NADH and FADH, that is 2.5 and 1.5 respectively, are the PO ratio of these two reducing equivalents. Now, you have seen in the first complex, the NADH and NADH, NAD and NADH redox couple, they are having minus 0.32 redox potential, and ultimately the electron is oxidized by the oxygen, where the redox potential is plus 0.82 plus 0.82. So there is a difference of plus 1.14 volt in the total chain. That is equivalent to 52 calorie per mole. Okay. Now each ATP, to generate each ATP, we need 7.3 kilocalorie, minus 7.3 kilocalorie. So to generate 2.5 ATP from NADH, we can capture 18.25 kilocalorie. So conservation of energy is nothing but 35.1%. Rest of the energy, that is out of 52 calorie, we can conserve only 18.25. Rest of the energy is dissipated as heat. But this process is not a wasteful process, because this heat we need for maintenance of the body temperature, and also for the electron transport chain reaction to go on. Okay. So only 35.1% energy is trapped, remaining 65% is lost as heat. This heat is necessary to maintain body temperature as well as to reactions of electron transport chain to occur. For two purposes it is used. Okay. Now, mechanism of oxidative phosphorylation. How does it occur? There are two mechanisms proposed. One is chemical coupling hypothesis by Edward Slater in 1953. In this, he was telling that there is during the course of electron transfer, the respiratory chain, there is series of phosphorylated high energy intermediate. Phosphorylated high energy intermediates are generated in the electron transport chain. But none of the high energy compound could have been demonstrated in the electron transport chain. So this hypothesis is out of order, nowadays it is not accepted because of the lack of evidence. So next is chemiosmotic hypothesis. This is placed by Peter Mitchell in 1961. It is widely accepted hypothesis. Here electron transport is coupled with translocation of proton that produces electrochemical and proton gradient across the inner mitochondrial membrane. The electrochemical in the sense that we have seen that translocation of proton is there in the intermembrane space. So in comparison to the matrix, the intermembrane space is more positive in charge. So there is a electrochemical gradient. Again, number of protons are there, more number of protons are translocated, so there is proton gradient. So two gradients are there, electrochemical and proton gradients across the inner mitochondrial membrane. ATP synthase present in the inner mitochondrial membrane utilizes this proton gradient for synthesis of ATP from ADP and inorganic phosphate. There are some requirements for synthesis of ATP according to Michel's hypothesis. There should be intact mitochondrial membrane. If it is not intact, then the proton gradient will be dissipated. There will be no more proton for passing through the complex 5 to generate ATP. Accumulation of high concentration of protons in intermembrane space, this is also required. What are the events occurring? Generation of proton gradients across the inner mitochondrial membrane and synthesis of ATP by ATP synthase, which is driven by proton pump. So here is the proton gradients. Number of protons are quite high than the comparison to matrix. So protons, they are coming through the complex 5 and there is generation of ATP from ADP and PI. Now let's see the machinery which is synthesizing the ATP. Two subunit, it is a knob and ball type of attachment is there. So this is the ball type and it has two subunits. The FO subunit and F1 subunit. F means oligomycin-inhibited subunit. This subunit can be inhibited by oligomycin. And F1 we'll see later on. So this characteristics of this subunit is FO is inhibited by oligomycin. It spans in the inner mitochondrial membrane. It is water insoluble, hydrophobic, that's why it can span the membrane because membrane core is hydrophobic. That's why it can attach in the, it can span the mitochondrial membrane, inner mitochondrial membrane. Then it has 10 numbers of disc-like structure. They are arranged in such a way that they form a channel in the middle which allows proton to pass on. So discs of C protein, these are the C proteins. They are 10 in numbers and while proton passes through this, there is rotation of this disc occurs during the passage of proton from intermembrane space, which in turn rotates the gamma subunit. This is the gamma subunit of F1 complex. This F1 complex has gamma subunit which is attached to the FO subunit. So when there is a rotation in this subunit, along with this FO subunit, the gamma subunit also rotates. It has a purpose, we will see later on. So it has three subunits, A, B, C. A which connects with the F1 subunit, then B, A through the B it connects with delta subunit of the F1. So C is 10 in numbers. They are arranged in such a way that they form a channel for passage of proton. Now F1 subunit This is the actual site of ATP synthesis. It is water soluble. That's why it hangs in the mitochondrial matrix. It projects into a matrix. It has 9 polypeptide chain, 3 alpha. Here are alpha. The green color 3 alpha subunits are there. Here, and 3 beta. Actually, it remains in the form of 3 alpha beta subunits. 1 alpha, 1 beta will form 1 subunit. So, likewise, 3 subunits are dimers are there. 1 alpha, 1 beta will form 1 dimer. Likewise, 3 dimers are there. So, there are 6 subunits here. Then gamma, it is attached with the FO and epsilon. 1 epsilon, then 1 delta. The alpha chain has binding site for ATP and ATP. This alpha chain, it has a binding site where the ATP and ADP bind, and beta subunit has a catalytic site. There ATP is formed from the ADP and PI. It requires magnesium ion for its function. Now here, if we see, consider only beta subunits. So beta subunits, here are the 3 beta subunits, here 1 beta subunit, here 1 and here 1. So beta subunits, it is the static portion, it doesn't move. And here is your gamma subunit, a part of gamma subunit. Transversal section we have given. So when the gamma subunits rotate, in beta subunit there is conformational change. Beta subunits, it has 3 different conformation. One is O form, it is open form. It has low affinity for the substrate ADP and PI. So when it is in O form, ADP and PI doesn't bind to this. Okay. Now another is L form. It is loosely, it has low affinity for substrate ADP and PI. So when it is in L form, this ADP and PI is loosely bound to the beta subunit. And another is T form. It is tight form. It binds substrate ADP and PI tightly and catalyzes ATP synthesis. Then in next, again this T form is converted into O form. Once it is O form, then the ATP is released. But ADP is not bound. It is releasing the ATP, and in the next moment it is transforming into L form. So here are 3 beta subunits. In a given time, there will not be 2 forms in 3 of the beta subunits. Means, in a given time, 1 beta subunit will be in O form, another will be in L form, and another will be in T form. So there will not be overlapping of these conformational change. Okay. So 3 beta subunits, though structurally similar but functionally not same at a particular time. Okay. Now suppose here. So here let's take from L. This subunit, it has loosely bound to ADP and PI. In the next, when this gamma subunit, when it rotates, it changes its conformation from L to T. Okay, it is not rotating, it is just changing the conformation. So ADP and PI, it is tightly bound here, and there is catalysis of the reaction and the ATP is formed. Just for understanding purpose, we are So if you consider this subunit, it has transformed into T form and there is ATP formation. Okay. In the next moment, this T form has, there is a conformation change to form O form. Okay. So once there is O form, the ATP is released, and in the next turn, the beta subunit, the same beta subunit, it assumes the conformational change of L form. Okay. Now it is the same 3 beta subunit changing with the rotation of the gamma subunit. Gamma subunit rotating due to the passage of the electron, proton, due to the passage of the proton through it. So for shifting, translocating 4 numbers of proton through gamma FO subunit towards the matrix will generate 1 ATP. Now, as we have seen, some of the reducing equivalents, they are produced in the cytosol. They are produced in the cytosol. But this mitochondrial membrane, it is not permeable to NAD or NADH. Means the reducing equivalent cannot come into inside the mitochondrial membrane. They are having some different mechanism of shifting these reducing equivalent inside the mitochondrial membrane, mitochondrial matrix. Here are the shuttle systems which are being used to transfer the reducing equivalents from cytosol to the mitochondrial matrix. Okay. Now these shuttles, they are given in short note. Okay. So there are mainly 2 shuttles, malate aspartate shuttle and glycerol 3-phosphate shuttle. Both the shuttles, they are given. Okay. Now so glucose in glycolysis, NADH is produced. Now this NADH can utilize 2 pathways, 2 shuttle system to get inside the mitochondrial matrix. These are tissue specific: liver, kidney and heart. They are using this malate aspartate shuttle. Other tissues, they are using glycerol 3-phosphate shuttle. In case of malate aspartate shuttle, the oxaloacetate, which is present in the cytosol, oxaloacetate, it is converted into malate with the help of cytosolic malate dehydrogenase. Cytosolic malate dehydrogenase converts oxaloacetate to malate using this NADH and H plus, so that the NADH which is produced by glycolysis in the cytosol is used up in formation of malate. Now this malate can cross the mitochondrial membrane, inner mitochondrial membrane. So it passes to the mitochondrial matrix, and there another set of malate dehydrogenase enzyme is there, that is called mitochondrial malate dehydrogenase. So that again produces oxaloacetate from malate, forming NADH, which is of our interest, because NADH we could not carry from the cytosol to the matrix. Now with the help of this reaction, we have indirectly transferred this NADH inside the matrix and it is regenerated. Now this NADH will take part in electron transport chain and produce 2.5 numbers of ATP. But this oxaloacetate, it is required to push it out from the matrix of the mitochondria. It is done how? It takes part in a transamination reaction. The amino acid for taking part is glutamate. So from the glutamate, the amino group will transfer to the oxaloacetate to form aspartate, and glutamate itself becomes alpha-ketoglutarate. Both the alpha-ketoglutarate is a keto acid and aspartate is a new amino acid. So both of them, they can cross this mitochondrial membrane and they come out from the membrane to the cytosol. Here again, they are cleaved. This aspartate, again they are taking part in transamination reaction, and aspartate is converted into oxaloacetate so that the cycle can continue, and alpha-ketoglutarate is converted into again glutamate. Okay. So in this way, the this malate aspartate shuttle is functioning for transferring the reducing equivalent NADH from cytosol to the mitochondria. In case of glycerol 3-phosphate, this glycerol 3-phosphate is a intermediary product of glycolysis. The dihydroxyacetone phosphate, it is also intermediary product of glycolysis. They are interconvertible, glycerol 3-phosphate and dihydroxyacetone phosphate. So NADH, this dihydroxyacetone phosphate, it is converted into glycerol 3-phosphate by the cytosolic glycerol 3-phosphate dehydrogenase, and it uses NADH. So glycerol 3-phosphate now it can cross the mitochondrial membrane, it goes inside the mitochondria. Again, by the use of by the help of mitochondrial glycerol 3-phosphate dehydrogenase, it is this dihydroxyacetone phosphate is regenerated, releasing not FAD, not NADH, but the FADH2. In the mitochondrial matrix, it cannot produce, this shuttle system cannot produce NADH again. It can produce FADH2. Now FADH2 take part in the electron transport chain and produce how much ATP? 1.5. So it produces 1.5. So this is the loss of this system, but because this tissue, skeletal muscle, brain and other tissue, other than liver, kidney and heart, they are using this system, so we have to accept it. Okay, there is, we are in cytosol, we have NADH, but in the mitochondrial matrix, using this shuttle, we are getting only FADH2. So these two shuttle systems are very important. Now another shuttle system is there, creatine phosphate shuttle system. Creatine phosphate, because ATP that is produced in the what is produced in the mitochondria, it is incorporated with the creatine to form creatinine phosphate. Creatine phosphate is a high energy compound. So creatine phosphate, it is transferred from the, it is carried, it can cross the mitochondrial membrane, so it comes out in the cytosol, and again it is carried to the site of use like muscle. Especially muscle needs creatine phosphate. At the time of muscle contraction, vigorous muscle contraction or maximum. And in the book also, if you read, so many things are there. You don't know which one is to leave, which line is to leave, and which line is to be remembered. So at least in the lecture you can know these things now. So creatine phosphate shuttle, it also comes as short note. Creatine phosphate shuttle. Now regulation of ATP synthesis. You cannot go on producing the energy. There should be some bar in producing energy. So there may be some limitation, there may be some requirement of producing energy. So availability of ADP is one. So if you cannot supply ADP, then you cannot synthesize ATP. No? So adequate supply of ADP is required. Need of ATP for all cells. When body needs more ATP, more synthesis will be there. So there should be a more need. There should be a need. It is only need based generation. Okay. So if body needs more ATP, then ATP is synthesized more. If less ATP is required, then it will synthesize less number of ATP. So once less number of ATP is synthesized, then where it will go? The energy, the food what we are taking, that will be converted into your triacyl glycerol and it will deposit as fat in our body. That's why in old days, when we need less number of energy, less amount of energy due to low BMR, the food what we take, it converted into fat, triacyl glycerol. It is deposited in the deposit tissue. That's why old people are fatty, no? So compartmentalization. Compartmentalization means, see, maximum of the reducing equivalents, they are produced in the TCA cycle. Okay, tricarboxylic acid cycle. So their enzymes are located inside the mitochondria, so that whatever reducing equivalents is produced, they can be adequately utilized in the electron transport chain. Okay. Then availability of the substrate availability also has to be there. Adequate foods and all these things, it should be there. During starvation, all the things, all the metabolic activities, it goes down. So an inadequate substrate will be there, inadequate ATP synthesis will be there. Now comes inhibitors of ATP synthesis. If it is said inhibitors of ATP synthesis, then you have to give all the inhibitors of electron transport chain and uncouplers. Okay, what is uncouplers? We will see now. And if you are asked for only uncouplers, you will mention only the uncouplers. Now let's see what is that. Inhibitors of ATP synthesis, they are the inhibitors of these complexes. Complex 1, complex 2, complex 4. And so in complex 1, inhibitors are chlorpromazine, rotenine is a fish poison, then amobarbital or amital is amobarbital is also called amital, then PRCD. They blocks the electron transfer in the complex 1. So once complex 1 is blocked, then there is no generation of proton gradient. There is no transfer of electron from NADH. So it is inhibitor of complex 1 and it inhibits ATP synthesis. Likewise, complex 3, these are British anti-leucate, also called dimercaprol, antimycin A, then naphthoquinone. For MCQ, very important. Okay, so naphthoquinone. These things, they block complex 2, complex 3. So there is likewise the electron transfer is blocked in transfer from the cytochrome B to cytochrome C1. Then complex 4, it is carbon monoxide, cyanide, azide, hydrogen sulfide. So MCQ, then again you will be asked why carbon monoxide is a deadly gas, or why cyanide is very much poisonous. Okay, you have to explain. Because carbon monoxide and cyanide, azide, hydrogen sulfide, they block transfer of electron from cytochrome A to A3 in complex number 4. So the proton gradient is not occurring due to blockage of electron transfer. Okay, that's why they are poisonous. Carbon monoxide, it binds with the reduced form of cytochromes, and cyanide, it binds with the oxidized form of cytochrome. Okay, so these are the inhibitors of electron transport chain. Inhibitors of electron transport chain also, they are inhibiting the ATP synthesis. Electron transport chain. Once they say inhibitors of electron transport chain, you have to say after this. Now, if they say uncouplers or inhibitors of oxidative phosphorylation, then comes the other things. Now, in case of this inhibitors of ATPase, means these are oligomycin. Oligomycin, it can inhibit the subunit FO subunit. Okay. Now, few words about the antidotes of cyanide poisoning. Antidotes or treatment for cyanide poisoning are amyl nitrate, sodium nitrite, and hydroxycobalamin. These nitrites, what they do? They convert hemoglobin into meth hemoglobin. Meth hemoglobin has a high affinity for the cyanide to form cyan meth hemoglobin. Cyan meth hemoglobin. Okay? Cyan meth hemoglobin leading to release of cytochrome oxidase. Okay? So, release of cytochrome oxidase. The cyanide, when cyan meth hemoglobin is formed, this cyanide has increased affinity for meth hemoglobin, so it releases cytochrome oxidase and binds with the meth hemoglobin. Cyan meth hemoglobin is not injurious to health, so here the cytochrome oxidase is released and it becomes again functional. Hydroxycobalamin converts cyanide to harmless cyanocobalamin. In vitamin B12, we know that another form of vitamin B12 is cyanocobalamin. So this cyanide, it leaves cytochrome oxidase and binds with this hydroxycobalamin to form cyanocobalamin. Okay? So by this way, it releases cytochrome oxidase and it is used as antidote. Now, uncouplers of oxidative phosphorylation. For oxidative phosphorylation to occur, the proton gradient is must. Uncouplers are those, and for generation of ATP, there is coupling of oxidative electron transport chain with phosphorylation. So uncouplers, what they do? They dissociate from from electron transport chain and oxidative phosphorylation. So they dissociate oxidation from phosphorylation. These are toxic and lipophilic compounds. They have to be lipophilic because they bind in the inner mitochondrial membrane. So they have to span the whole membrane so that they can transfer electron or a proton or positive charges from the mitochondrial space to mitochondrial matrix. Make intermitochondrial membrane, inner mitochondrial membrane permeable to protons. As such, it is not permeable. Because of that, proton gradient is there. If proton has to pass through, it has to come through the complex 5 so that ATP is generated. But uncouplers, what they do, they are acting like a hole in the membrane, so that through that hole, the proton, it passes from the intermembrane space to the mitochondrial matrix. So ATP generation is not there. Thus, energy prevent formation of proton gradient in the intermembrane space. Thus, energy produced by the transport of electron is released as heat, not ATP. So ATP is not formed, proton gradient is dissipated, and heat is released. But this generation of heat is many a time it is necessary. Some of the uncouplers are 2,4-dinitrophenol, dinitrocrazole, pentachlorophenol, chlorocarbonyl cyanide, phenyl hydrazine. So these are the uncouplers. Remember these things. They will give in MCQ. They will put one, identify the uncoupler. They will give 2, 3 names, 4 names of that molecule. One will become here. Okay. Now, physiological uncouplers. Physiological uncouplers, they are, so the important is thermogenin. Important physiological uncoupler is thermogenin. Thyroxine also, in high dose, it acts as physiological uncoupler. Long chain free fatty acid, it also acts as uncoupler. Then high level of bilirubin, it also acts as uncoupler. Okay. Now, thermogenin, there are some adipose tissue. They are called brown adipose tissue. It is present in the hibernating animals. Then it is present in the small children, where body temperature is needed to be maintained. So these thermogenin, they uncouple oxidative phosphorylation from electron transport chain, and proton gradient is dissipated as release of heat. But not all. There is heat production as well as ATP generation is there. So it is important for kids. So significance of uncouplers. Here you may have short note as brown adipose tissue. You may have short note as uncouplers, physiological uncouplers. So in both the cases, answer will be, you can write the same answer. So uncoupling of respiration electron transport chain from oxidative phosphorylation under natural conditions assumes biological significance. The maintenance of body temperature is particularly important in hairless animals, all newborn mammals including human, hibernating animals, and the animals adapted to cold. These animals possesses a specialized tissue called brown adipose tissue in the upper back and neck portion of the body. The mitochondria of brown adipose tissue are rich in electron carriers, are specialized to carry out oxidation uncoupled from phosphorylation. There will be oxidation, there will be electron transport, and it is uncoupled with the phosphorylation. This causes liberation of heat when fat is oxidized in the brown adipose tissue. This is important for maintaining body temperature. Brown adipose tissue may be considered as a site of non-shivering thermogenesis. Usually, when it is very cold, we used to have shivering, no? The purpose of shivering is to generate heat, so that body temperature is maintained. So in brown adipose tissue, it causes heat generation without shivering. That's why they are calling it is non-shivering thermogenesis. The presence of active brown tissue in certain individual is believed to be protect them from becoming obese. See, so many people are there taking a lot, no, but not becoming obese because they are having more number of brown adipose tissue. The excess calorie consumed by these people are burnt and liberated as heat instead of being stored as fat. Physiological uncoupler or thermogenin. Thermogenin itself is maybe given as a short note. Thermogenin is a protein present in mitochondria of the brown adipose tissue. This tissue is rich in mitochondria. It is present in all newborn mammals, including human. Brown adipose tissue mitochondria are similar to other mitochondria, except that they contain an additional protein called thermogenin. Thermogenin acts as channel for migration of protons from intermembranous space into matrix. This uncouples the oxidative phosphorylation, leading to release of free energy as heat. This is needed to keep the newborn baby warm and maintain the temperature. This much I have written because I want you to these things to be mentioned in the short note. Okay, so because in book so many things are written, which how much to write, what is to write, that you may confuse. Brown adipose tissue, thermogenin, physiological uncoupler. One of the out of these two you will get short note. Ionophores. These are the molecule that allows passage of these cations through the intermembrane space. The term ionophore is used to collectively represent the lipophilic substance that promotes the transport of ions across the biological membrane. So far we have mentioned those things, these dinitrophenol, crazole, then thermogenin. These are the all the uncouplers described are in fact proton ionophores. Proton ionophores, they allow passage of proton. But the antibody valinomycin, germicidin A, gramicidin A, and nigersin act as ionophore for potassium ion. Both these compounds are also capable of dissipating proton gradient across the inner mitochondrial membrane and inhibit oxidative phosphorylation. So, if this potassium ion is allowed to pass, the electrochemical gradient is dissipated. So, there also the ATP will not form. Oligomycin inhibitors of oxidative phosphorylation, other inhibitors of oxidative phosphorylation. Oligomycin, it inhibits complex 5. It is antibiotic, prevents the mitochondrial oxidation as well as phosphorylation. It binds to the enzyme ATP synthase and blocks the proton channel. It thus prevents translocation of re-entry of proton into the mitochondrial matrix. Due to this, the proton gets accumulated in the high concentration in the intermembrane space. Electron transport ultimately stops since proton cannot be pumped out against. So many protons are there inside. So you cannot push any more proton inside, and this proton also cannot pass through the matrix because oligomycin has blocked the complex 5, this C discs of the FO unit. So that also stops the synthesis of ATP. Attractyl site, this time it has given in the exam. Attractyl site, we have seen that ADP-ATP translocase. Okay. So attractyl site, it blocks ADP-ATP translocation. So there is ADP cannot go in, ATP cannot come out. So again, the substrate is not available, so ATP cannot be produced. So here, this is a plant toxin, inhibit oxidative phosphorylation by an indirect mechanism. Adenine nucleotide carrier system facilitates the transport of ATP and ADP. That is ADP-ADP translocase, and thus blocks the adequate supply of ADP, thereby preventing phosphorylation. Okay. So this is in short the proton gradient is created in the intermembrane space that is passed through the complex pi, and with the rotation of the FO subunit of the complex pi, there is synthesis of ATP from ADP. This ATP needs to translocated out from the mitochondrial membrane by translocase, ATP ADP translocase, and the phosphorylation, electron transport and phosphorylation, they go on in a coupled manner. So these are the questions that will be, that is frequently given in the university examination. Okay.